Greetings everyone, and today we are going to discuss about the structure of bacteriophage. What are bacteriophage? Viruses which infect bacteria are known as bacteriophage. These are uh, bacteria means bacteria phage eater. These are bacteria eater. These are organisms which are basically not organisms. Actually, viruses are acellular life. So uh, these are organisms which feed on bacteria as well as cyanophages. Those phages, those viruses which act on, you know, cyanobacteria as well as when it comes to microphages. Microphages are those which uh, feed on microphages are those not about macrophages. Macrophages are those which feed on fungi as well as uh, you know phycophages are those which feed on phycophages are those which feed on algae, and as well as zymophages. Zymophages are those which feed on yeast particularly, and uh, you know yeast, and phytophages are those which feed on plants. So now we're going to study the structure of bacteriophage. Bacteriophage are divided into two major types, which includes the T even phages. Examples of these include T2, T4, T6. Most of the bacteriophages contain double-stranded DNA. There are some exceptions of virus bacteriophage which do not contain double-stranded DNA. Let us first discuss the common things. Here we are having T-even phages and T-odd phages. T-even phages, T2, T4, T6, and T-even T-odd phages include T3, T3, T7, and T1. These are the T-odd phages. These are both T-even and T-odd phages are double-stranded DNA phages. Moreover, when it comes to the phages, other sort of phages, there's lambda phages, lambda bacteriophage. These bacteriophage are double-stranded DNA viruses. Double-stranded DNA viruses are double-stranded DNA viruses. Alongside which, when it comes to an exceptional case here is Phi into EX174 bacteriophage. This is a bacteriophage which is having a single stranded DNA, whereas T even phage, T odd phage, and lambda phages are having double stranded DNA. Most of these are having double stranded DNA, and apart from that, bacteriophages basically uh, are divided into two types, which includes bacteriophage which undergo lytic cycle, and those which undergo lysogenic cycle. Those which undergo lytic cycle are known as lytic phages or virulent phages, because these are the phages which enter inside the host cell and infect the host cell alongside kill the host cell in order to produce and replicate their progeny. But lysogenic phages are those phages in which they infect the host cell but do not kill the host cell. Rather, they incorporate with the host cell genome and become a part of the host cell genome. So these are known as temperate phages, prophages, or lysogenic phages. And the bacteria in which lysogenic cycle occurs is known as lysogenic bacteria. These are the things we are just going to discuss now. Coming to the basic intro of how does the structure of bacteriophage look like. So the structure of bacteriophage first and foremost consists of three major parts. First includes the head, neck, and tail. Head is polyhedron, which is made up of it is a icosahedral shape which is having 20 equilateral triangles as previously mentioned, which is arranged in a polyhedron shape. Moreover, there is the presence of a neck which is linear, as well as we are having a tail which is present over here, is which is known as helical tail, which is basically it's having a hexagonal, tail is in the form of a helical structure. The nucleotides are arranged in helical structure, so there is a rod shaped tail, as a result of which this forms a rod shaped structure of the tail. When it comes to some other structures found over here, let us discuss them one by one. First and foremost, there is a capsid head. Capsid head basically, as discussed, capsid consists of, here the structure is quite complex because it is, divided into the head part, tail part, as well as the tail fibers. So here when it comes to the tail part, head part particularly, head part here involves head part here involves first and foremost, the capsid is made up of capsomeres, and in bacteriophages around 2000, 2000 or more than 2000 capsomeres are present, apart from which when it comes to their viral genome includes a double stranded DNA, which is linear DNA, not a circular DNA. Moreover, some other specific features which I would like to highlight over here are about the second part, which is the neck. Neck part basically is just like a short tube which connects the tail with the head part, and that part basically, as you can see in the diagram as well, this neck part is having several, it is having a site through which the head and tail are connected with each other. So this is a site which contains specific proteins through which the are head and tail are connected with each other, and this part is known as the collar. Collar is the part, and sometimes we can also commonly call neck as collar, because there's not much difference between both of them, because collar mostly covers most of the neck part. Apart from which, side by side from the neck, from the collar, there are several arrangement of several, pin-like structure, which is known as the viscous. As you can see the diagram, maybe it would be mentioned or not. There are several pin-like structures which are known as whiskers. Now, down towards the sheath, down towards the neck, there is a present of the tail. This complete structure, elongated structure, is known as tail, which is basically rod shaped, and that tail, which is rod shaped, is known as uh, the tail part, and this tail part is having two major sides. First side is the inner tail. I mean, as you can see in the diagram, let me just try to draw an diagram which can help you understand how the structure looks like. Here there is a tail part. The tail part is divided into two parts. The inner hollow tube is present of the tail, inner hollow tube of the tail, which is basically non-contractile in nature, whereas the outer contractile sheath is there around the tail part. Outer contractile sheath is present around the tail part, which is known as the tail sheath, which is known as the tail sheath. This tail sheath basically is contractile and thus can contract in order to inject or the host penetrate the viral genome within the host cell, as a result of which the it helps in penetration. Moreover, down towards the tail, there is the presence of a base plate. This base plate contains specific types of proteins, uh, which, as well as it contains a special enzyme known as lysozyme, which is used for the lysis of the bacterial cell wall components, as well as in order to make several minute holes within the bacterial cell wall. Moreover, within the base plates, it is having specific proteins for itself, as well as several tail pins. These tail pins are basically having a characteristic of recognizing the particular. They have a characteristic to recognize which type of cell they are supposed to infect. For suppose if there is bacteriophage, then the virus bacteriophage, if they for suppose if there is a bacteria coming, then the bacteriophage is then able to know this is my host, I need to attack this particular bacteria. So at that moment, tail pins are the part which are, irreversible, which have the reversible, I mean, they have the characteristic to recognize which part they need to which bacteria they need to bind with and penetrate and attack. Moreover, one more thing, that bacteriophages mostly attack E. coli bacteria. So uh, these are known as coliphages as well. One more thing over here that comes in my mind is these fibers, these tail type, these, legs of bacteriophages. These are basically fibers of bacteriophages. These bacteriophage fibers are basically known as tail fibers, or we can also call them as caudal fibers. These caudal fibers are attached to the base plate, and down towards the caudal fibers, here there you can see there is the presence of, at the ending part of the tail fibers, caudal fibers, there is the presence of, here there is the presence of specific receptor binding protein which binds to the receptor binding site. It contains basically a receptor binding site which binds to the receptor binding protein which are present on the surface of the bacteria, so that it can form a mutual affinity with the receptor binding protein of the bacteria, as a result of which when it binds with the receptor binding protein of the bacteria, it is then able to bind with it, and thus helps in adsorption process, after which penetration occurs. And hopefully we are going to discuss all this stuff into the lysogenic and lytic cycle. So this is how the basic structure look like, and we can also call these spikes as you know uh, these tail pins as spikes. Depends on our wording, as well as examples of these phages include lambda phages, T4, T7, uh, P1 phage, M13, lambda phages, T4, T7. I mean, many examples are given. Moreover, coming to the lytic cycle basically here involves the following five steps. The first step is quite simple. Uh, which is basically the attachment of the phage. When the receptor binding site of the caudal fibers which are present over the bacteriophage attach with the receptor binding site over the surface of the bacteria, which cause to help in the process of adsorption and attachment. As a result of helping in the process of adsorption and attachment, there is, as a result of process of adsorption and attachment, it causes the host bacterial cells receptor binding site to attach with the receptor binding protein of the caudal fibers of the bacteriophage, as a result of which adsorption process occurs. Now in this case, the receptor binding site of the bacteria is having a mutual affinity with the receptor binding protein of the bacteriophage, as a result of which there is a mutual affinity, and they are able to form a sort of complex in order to adsorb, attach, or bind. Now, after the binding, the bacteriophage uh, s- starts to release its enzyme, which is known as lysozyme, from the base plate. Uh, from the base plate, it releases a lysozyme enzyme. Using its tail pins, it releases the lysozyme enzyme, which causes lysis of the bacterial cell wall components, as well as makes minute holes. As a result of which, what it actually does is that it contracts itself. It contracts its a contractile sheath, tail sheath. As it contracts its contractile outer tail sheath, it causes the inner hollow tube to the inject within the host cell's cytoplasm, in such a manner that, let's suppose if we are having uh, you know if this is a virus, oops, let's suppose if this is a bacteriophage, and here I'm just drawing its clone. I'm just drawing another bacteriophage. It's a bit irregular, but you can better understand it. So here, as the bacteriophage tends to attack, this bacteriophage first and foremost tends to contract its contractile sheath, as a result of which the hollow tube comes downwards and comes over in contact, comes it injects, just like an injection, as we are supposed to inject the needle of an injection within the person's skin. In the same manner, this contractile sheath contracts, as a result of which the inner hollow tube is injected, penetrated within the host cell cytoplasm. As a result of injection, it causes the viral genome to be injected within the host cell cytoplasm. Of course, now there's also the role of tailpins here. That how does tailpin help in this process? Basically, when the contractile sheath contracts, the tailpins basically, you know, uh, penetrate into the surface of uh, penetrate into the surface of the host cell, as a result of which the as a result of which the contractile sheath contracts and it is able to release a viral genome within the host cell. Now, coming to the next part over here, that what is the next step, which includes the biosynthesis or replication. This biosynthesis or replication occurs in such a manner. Basically, bacteriophage, the bacteriophage which we are talking about is T4 phage. We are specifically discussing the T4 phage. So T4 phage here, particularly, which we are going to discuss, T4 phage. Now, what it does is that it has a gene. It has gene for synthesis of endonuclease enzyme, such that when it synthesizes its endonuclease enzyme, this endonuclease enzyme causes the degradation of the host cell genome, as a result of which the metabolic machinery of the entire host cell comes under the control of the viral genome, as a result of which now big viral genome controls the entire cell and starts to form its desired protein. For suppose it starts to form its structural proteins, as well as it utilizes the degraded host cell genome to uh, be directed by its own self to be utilized in its own viral genome synthesis. Moreover, it also does replication of itself in order to form its multiple copies of its viral genome, as well as forms mRNA in order to form its structural proteins. And in such case, it also, you know, forms three sets of proteins. Three sets of proteins are particularly formed, which are of benefit to it, which includes the head part protein, tail part protein, and fibers part protein. So these three parts of proteins are formed. These sets of proteins are formed, and once this replication occurs, synthesis occurs, phage proteins are made. It then moves on to the next step, which includes the assembly, or we can also call it as a maturation. In this process, the structural proteins as well as the viral genome are assembled together according to the genetic instructions which are contained within the bacteriophage. One more thing that is present over here is that it has, you know, some bacteriophages are also having gene regulatory system. For suppose if we talk about bacteriophages which undergo lytic cycle, let's take example of lambda phages which undergo lytic cycle are having the characteristic of gene regulatory system, such that they can turn on and off their genes by utilizing inhibitor and enhancers. Now in this case, what we are going to do is, have the genetic material according to the genetic instructions, the assembly of the structural proteins and structural viral genome occurs, due to which the new phages are formed. Now, the complete infectious particle which is released from the infected cell is known as a virion, which is also known as virion or mature virion. Which now, once the virion is formed, the bacteriophage starts to form the lysozyme enzyme, which causes a lysis of the bacterial cell wall component and makes minute holes in the cell wall. As a result of lysis of the bacterial cell wall, it causes the plasmolysis of the bacterial cell, due to which its cytoplasm is shrinked, and thus, as a result of shrinkage of bacterial cell cytoplasm or plasmolysis, the bacteriophages are also released from the cell and are ready to infect other cells. This is a complete cycle. When it comes to lysogenic cycle, lysogenic cycle is quite similar, except the fact that here we are have it basically incorporates forms a relationship. Basically, in lytic cycle, there is a slave relationship. That master is bacteriophage and slave is bacteria. Whereas in the lysogenic cycle, there is a host guest relationship. Just like a guest uh, stays at the host's house, uh, bacteriophage remains within the bacteria's cell for many years and within its progenies until unfavorable conditions arise. Let's discuss. So, lysogenic bacteria, as I previously told, are those which are having lysogenic cycle in them. So when you know lysogenic phages or temperate phages or lysogenic phages or temperate phages or prophages enter inside the cell. They basically, the first processes are simple. That first process is binding and penetration, same as lytic cycle. After that, what they actually do is incorporate their genome with the host cell's genome. As a result of integration, it causes uh, to form a new genome which is known as prophage. Now here, one more thing, that genome becomes virus when it enters the host cell. Genome becomes a virus when it enters the host cell. Now this is known as prophage, as mentioned in the diagram as well. This is the viral genome, as you can see in the cycle. First, there's integration. The prophage excites from the bacterial chromosome and enters the lytic cycle. Lysogenic uh, phage DNA circularizes, enters lytic or lysogenic cycle. Now phage DNA integrates within the bacterial chromosome, and once it is integrated, now lysogenic bacterium reproduces normally, just like a host and guest relationship is there. So it remains within the bacteria for many generations, and remains within a lot of progenies until there is any unfavorable circumstances, which includes mutagenic chemicals or ionizing or ultraviolet radiations, or increase or decrease in pH, or increase or decrease in temperature, which can lead to unfavorable circumstances, the lyso due to which the virus, n- after facing these unfavorable circumstances, come out of its lysogenic cycle and then convert back into its lytic phase. One more thing, that as I previously mentioned, that here, uh, as the bacteria is having its lysogenic cycle going on, its gene for replication are turned off. Its capability to destroy or perform the lytic cycle is not destroyed at all. Only there is the close switch off of the gene, which means here there are genes for lytic cycle, but genes are turned off by the inhibitor present within it, as these are having gene regulatory system as well. So as these are having gene regulatory system, they are just supposed to turn the gene off, and as they turn the gene off, this turning of gene off leads to lysogenic cycle. Now when they face unfavorable circumstances, again lytic cycle occurs, in which the gene is turned on again, and at that moment they perform lytic cycle. And this process in which the lysogenic cycle breaks and lytic cycle starts, then the bacteriophage is known then this process in which bacteriophage undergoes lytic cycle is known as induction. One more thing, that here lytic cycle occurs again. There is production of several progenies of the virus, I mean the bacteriophages, through the following five steps of the lytic cycle. It causes to infect other types of cells and settles within other bacteria, so that it can incorporate with other host cells genome and perform its lysogenic cycle within other cells. So in such case, this cycle goes on, and this is all about this video. Hopefully, we'll move again and meet in another one.